or for your kidneys. We're going to talk a lot about it's like the liver. You know, the liver has a couple different functions. It functions with making, you know, proteins for your plasma, detoxifying drugs and alcohol, digestive function. So your kidneys do a whole host of things. Today, we're going to primarily be talking about what your kidneys do in regards to filtering the blood. We've talked about some of their other roles there. You know, it's the site for erythropoiesis. Not the site for, I'm sorry, it's the site that we make EPO, erythropoietin, that helps with the production of your red blood cells. Sorry, I'll go back. Why isn't this going away? I can never figure that out. All right, now it should go away. Why? What am I doing wrong, y'all? There we go. Make that top thing disappear. Okay. So this chapter, we're going to be focusing on the filtration process of your kidneys. Essentially, they function as a big filtering mechanism for clearing out your blood. And so you can see on the whole host of things what they're going to do in the beginning. Elimination of metabolic waste, you know. So of course we know metabolically active tissues are going to be creating waste products. Your car creates waste products through carbon monoxide. We create waste products. We just talked about it in chapter 26 with our feces. So we're going to see now how with our kidneys, they're going to get rid of a whole host of these waste products. We're going to also, back in the beginning of the semester, we talked about how with the endocrine system, how certain hormones, right, will help regulate electrolyte levels. And so we're going to see that the kidneys are one of the organs that are going to play a role in that. Sodium, potassium, calcium is listed up there, right? But we'll see how it's also going to regulate, right, your pH levels of your body. Briefly talked about it in lab, how if you have too much carbon dioxide, right, floating in your blood, what that does to your respiration rate. But we're also going to see what it's going to do with the filtration of the functioning of your kidneys and getting rid of either hydrogen ions. And so your hydrogen ions are associated with the acidity, right? So the higher the hydrogen ion concentration, the lower the pH and the more acidic a solution is. But also there's the opposite of that. That's bicarbonate, by the way, HCO3. And bicarbonate is going to be, right, help with buffering, but what happens when your blood is too basic or too alkaline? Then we got to dump off and get rid of that bicarbonate there. Blood pressure, your kidneys, believe it or not, are one of the main organs that regulate blood pressure. Because, remember, one of the factors that influences blood pressure is blood volume. So if we can affect blood volume, we can affect blood pressure. And then, of course, we'll talk about a little bit what it does, all right, with hormones and drugs, okay? It's going to be dumping. Usually the two organs that you're concerned about, is anyone here going to be going into anything that has to do with pharmacology? Well, drugs, right? When you essentially are going to be taking drugs, there's organs that are going to metabolize the drugs. It's usually the liver or it's going to be the kidneys, right? And so we're going to see how the kidneys handle that, especially with some of the pharmaceuticals that you take. All right, we've seen pretty much all of these other functions here. Formation of calcitriol, which is what? Vitamin D. Yep, the active form of vitamin D. So we now know, all right, or we've known ever since chapter 6 when we learned about the skin, right, where vitamin D is first made, and then it has to go on a nice little journey. It goes to the liver and it gets converted into calcidiol. Still not the active form. Then it makes it to the kidney where it gets converted to calcitriol. And then we have, all right, erythropoietin production there. So when you have low, all right, blood oxygen saturation, we want to increase our red blood cell count. So erythropoietin gets released. And so that helps stimulate the red bone marrow in your, all right, bones, okay, to produce more red blood cells. Now, the last part, and I always wait till this chapter to even mention this. Up until this point, when you've heard me mention gluconeogenesis, it's always been liver, liver, liver. But in times of emergency, all right, the kidneys will help out with gluconeogenesis, which is nice. It's kind of like our fallback, our backup. So to recall, gluconeogenesis is the creation of glucose, right, from a non-carbohydrate source. So amino acids, fatty acids, things of that nature, right? Because that is one molecule that you want to maintain, all right, adequate levels of is glucose, because that's your brain's crack, all right? It loves glucose, right? In fact, we're going to talk about the glucose sparing effect, in which other tissues will forgo an opportunity to utilize glucose so your brain can have it, because your brain's a prima donna, and it always has to be the brain's way, all right? So let's talk about the 3 fluid names for the production of urine. What does that mean? Okay, so when you start to filter things out of your blood, right, whatever that stuff that gets filtered out is going to change names 3 times until eventually it reaches urine status, that it's even going to be part of the urine. All right, so there's 3 names: filtrate, tubular fluid, and urine. We talked a little bit about that in lab on Tuesday. So filtrate is what we filter immediately out of your blood in the glomerulus. It's going to flow into the capsular space, or Bowman's capsule, out there in the renal corpuscle, and then it's going to move into the proximal convoluted tubule, which it changes names there. So know what we call that fluid that gets filtered out of your blood initially. That's filtrate. Makes sense, right? We filter out the filtrate, right? And so that's going to be stuck. Or I shouldn't say stuck, but that's going to be captured in the capsular space there. You cool on that? And it's just going to be, right, when you're filtering out the blood, it's going to be the stuff that can move through this filtration membrane. And we'll discuss what that is. So you shouldn't see, like, is it normal to see red blood cells in your urine? No. White blood cells? No. Right? Large protein molecules? No. Right? So we're going to be filtering out certain specific things, and we'll discuss what those things are, right, in this chapter here. All right. Once it moves from the capsular space, or Bowman's space, into our tubular system, right, into that's the second part of the nephron, right, which is the glomerular, the tubule system there, we change names and call it tubular fluid. And so it's going to be known as tubular fluid for a lot of this journey through the nephron. You can see all the parts and pieces. So it's going to be known as tubular fluid through the proximal convoluted tubule, the nephron loop, right? Our distal convoluted tubule, and even as it goes into the collecting tubules and moves into the collecting ducts, it's still tubular fluid. The only time, right, that it is going to be known as urine is when it drains out of the renal papillae. All right, and that's going to, and we labeled what that is. That's that bottom part, that kind of cup at the bottom part of the renal pyramids there. So once it passes through the renal papillae, then it's known as, all right, urine. And then it's going to be known as urine for the rest of its time in the urinary system. Okay, so it'll move, all right, into the minor, major calyces and the renal pelvis, down through the ureters, into the bladder, then out through the urethra. All right, so keep that in mind. You want to know it's going to be referred to as tubular fluid here. And then finally here with urine, it's going to be known as urine through the rest, or the distal part of the urinary system there, which is going to be on the other side of the renal papillae here. So as it moves into from the minor calus to the major calus to the renal pelvis there. And then from the pelvis to the ureters, from the ureters to urinary bladder, and then out through the urethra. And we'll actually today in lab finish up discussing the urinary system from the renal pelvis all the way up to the exit of the body there. So some of this stuff might look unfamiliar to you. I doubt that it is. Don't worry, because after today's lab, you're going to be masters. All right, thoughts, questions, concerns? No? All right. If you need me to speed up, I will. So you can't really see this. Let me zoom in a little bit. This is just a nice picture kind of showing you the name, all right, of that fluid in its specific area there. So here, filtrate in only one spot. There inside of Bowman's capsule, or the glomerular capsule there. Right, and then as it moves into the tube system here, so all this through the proximal convoluted tubule, down through the nephron loop, into the distal convoluted tubule, through the collecting tubule, even in the collecting duct here, all of that, right, that is going to be the tubular fluid. And once it passes out through, right, the renal, the papillary duct, or the renal papillae here at the base of that renal pyramid, then it's known as urine. So as it passes through, then you can see where we refer to it as all right, urine here through the minor major calyx into the renal pelvis here, into the ureter, into the urinary bladder, and then out through the urethra. Cool. So we'll go through that. You'll get the hang of it. All right. Just don't want you to be hung up on, the name there and not be aware. All right. Now comes the fun part. Now we get to talk about how your pee is made. Then we'll talk about what influences urine production, what can increase it, what can decrease it, right? How we're going to maintain or increase or decrease blood pressure there. So there's 3 terms that are listed up here on the screen. All right. Our 3 terms are filtration, resorption, and secretion, sometimes known as excretion. I'm going to define all those for you, kind of explain what it is. The first one is the easy one, glomerular filtration, or just filtration. So this is the initial process. This is what's going to happen when blood is moving through the glomerulus. Remember the glomerulus. What's the glomerulus? Yeah, it's that balled up area, all right, or structure of capillaries. Okay, so you have the afferent with an A, arteriole, that moves into the capsule there, into Bowman's capsule, and you'll see just these loops of capillaries. This guy, right? I know I got a picture. There it is. Okay, so here you can see there's the afferent arteriole moving in, all right, and you have these loops of capillaries there. So that is where we're going to see the first step here. This is where we're going to see substances in your blood plasma. And so we already know what's in the blood plasma because we learned about it in chapter 18. Primarily water, okay? But we're going to see some other substances in there, right? You're going to see ions in there. Think of things that are really, really small, okay? Very small proteins. Ideally, you don't want to have proteins leaving, right? But you're going to have really small substances, hormones, not ions, I already said that, glucose, for example. So all of these things are going to be filtered out into the capsular space there, okay? They all reside in the blood plasma there. And so basically, okay, because we've studied in other chapters, right, pressures, okay? We know fluid pressure is known as hydrostatic pressure. So that still exists. This is no different, right, than any other blood vessel. You have hydrostatic pressure, and that's that pressure that the fluid inside of the capillaries, inside of your blood vessels, exerts on the walls of that blood vessel there, right? So that's the main thing. So this statement here, due to pressure differences across the filtration membrane, that's what's going to drive fluid from your blood into the capsular space. So obviously, we don't even have to learn all the fancy equations and whatnot. The pressure inside the capillaries has to be greater than the pressure in the glomerular space there, inside the Bowman space, okay? Because what's the fluid going to go? From an area of high pressure to low pressure. So if it's higher in the capillaries and lower outside of the capillaries, inside of the Bowman space there, the fluid's going to move into Bowman space. And we're going to talk about things that influence that. So once that fluid moves into Bowman space there, it's now called filtrate, right? So the second term there is resorption. And so we call it tubular resorption because it's going to occur in the nephron tubular system there. And so we're going to see now, this is where we get really, really, how do I want to say this? Where we go back to these processes that you learned way back in chapter 4. We're going to be talking about, we're all familiar with osmosis and diffusion, right? Because they're very close to being the same thing, except one involves water and the other one involves solutes. All right, and then we have our active transports. You remember, osmosis and diffusion are passive processes. They don't cost us any ATP, right? So that's nice. Our active transport processes do, and we'll discuss that. But essentially what we're going to see is we're going to see these materials essentially leaving that tubular system and going back into your bloodstream. Right? And that's influenced by certain conditions that might be occurring in your body. Right? If you're very hydrated or if you're very dehydrated, is going to determine in which direction, all right, that water is going to move, for example. Yeah. Oh, wait, this is vodka. All right. And I got my coffee. Too much creamer, though. I wish. No, no. I mean, no, I've never. For those that are listening on the recording, never. No. So anyways, when we, again, just quickly review, when we move into the tubular system, keep in mind, one, the name of the fluid changes from filtrate to the tubular fluid there. And then things are going to move from the tubes back and forth into the blood vessels that surround these tubes. And so you can see the list of processes that will occur with, all right, our passive processes of diffusion and osmosis, and then our active transport, which we'll talk about, okay? We'll talk about. So one of the things that you're going to see that these two things, we haven't really talked about it yet, we will talk about it in lab today, are these two capillary beds. One's called the peritubular capillaries. Look at that word, peritubular. So these capillaries pretty much surround your tubular system. So they're going to be enclosed around your distal convoluted tubule, your nephron loop, your proximal convoluted tubule, your collecting ducts and collecting tubule there, okay? So you're going to have all these capillaries around there, right? And then the vasa recta specifically is going to be around the nephron loops, all right? But the peritubular capillary is going to be mostly around the distal convoluted tubule, proximal convoluted tubule, all right? So we're going to see, right, these solutes and these different substances moving from the tubular fluid into your blood. And in some cases, they might move back out of your blood back into the tubular system. And we're going to discuss all that. But what we're going to see is, one, we want to get rid of the waste products, right? Okay, so there's nitrogen-based wastes that we want to get rid of, i.e. urea, which is a big component of urine there. And then if we have too much of something, we discussed that, right? For example, if you have too much calcium in your blood, if you are hypercalcemic, we want to get rid of calcium somehow. One of the ways that we can do it, aside from the kidneys, do you remember one of the ways that we can lower our blood calcium levels? Calcitonin. Okay, great. You're going there. So you're talking about that hormone. No, that's great. So what does calcitonin do? Yeah, well, it puts calcium into stuff. Specifically, what's a big reservoir of calcium in your body? Your bones. Yeah. So it's going to stimulate the osteoblast to build more bone matrix and stick the calcium into the bone matrix there. But it's also going to tell, all right, your kidneys to dump calcium into your urine. And so we're going to see that. So when you have excess solutes, depending on what it is, that's going to stimulate the direction of where, all right, these solutes are going to go. If you have excess solutes, you're going to dump it into urine, you're going to pee it out. If you have a shortage of solutes, if you're calcium deficient in your blood, for example, hypocalcemic, what's the other hormone that's going to be activated? Yeah, PTH, parathyroid hormone. So parathyroid hormone is going to then liberate calcium from your bones. It's also going to work synergistically with calcitriol, right, in absorbing calcium out of the digestive tract, huge there. But what else is it going to do? It's going to decrease, right, the excretion or secretion of calcium in your urine, right? So we're going to learn about that. So all the stuff that you kind of, when we led up to this all semester long, we didn't really get into it. Now we're going to see some of the specifics, right, of how the kidneys are going to make this happen. All right, so the last term there is called tubular secretion. Sometimes you'll hear me say excretion. It's the same thing. So what we're going to see now is the movement of more of these solutes. So it could be sodium, potassium, calcium, all sorts of stuff. But now we're going to see how active transport plays a role in that. And basically, when you're secreting something, you're going to be moving it from your peritubular capillaries and the vasa recta capillaries. We're going to be moving that stuff out of the blood back into the tubular fluid, and then we're going to pee it out. Right? So usually that's going to be if we do have like an excess of something, like if there's too much bicarbonate, okay? If your pH for your blood is too high, okay? If it's more alkaline, you're going to pee out bicarbonate. If your blood levels your pH are too low, like if it's like 6.8, 6.5, right? That means you have too much hydrogen ions floating around. We got to pee those out. So we'll go through this. We'll get into the specifics here. Probably not today. I love this picture. This is figure 2410. It's very, very helpful, right, to kind of see what I'm talking about. So you can see the green arrow here matches up with the green box at the top here. So the glomerular filtration just shows you, right, stuff leaving your blood and going into the capsular space there. And then it's going to move from the capsular space into the proximal convoluted tubule. And so if it's purple, right, that's showing you tubular resorption, which means stuff is going to be moving from the tubes into your capillary beds that surround the tubes, depending on the location, right, which is either going to be the peritubular capillaries or the vasa recta, right? So it means it's going from the tubes to the blood. That's resorption. And then we've got this kind of tan colored box here to match with the tan colored arrows there. Tubular secretion represents when stuff moves out of your blood into the tubular system there. Okay, and that's going to be your tubular secretion. So in some situations, depending on what's going on with the body, which we'll go through some certain scenarios here, we'll kind of discuss that. But I think this is a great picture here to kind of see, right, the direction of the movement of solutes, of water, of whatever, seeing those arrows there. So this is very helpful, at least in my opinion, figure 2410. Very good. All right. So let's start way at the beginning, right, with our filtration membrane, because we're going to be moving stuff out of your blood, out of the circulatory system into our nephrons there. But we don't want to move everything out of the blood, only certain things out of the blood, okay? So if you look at the first description there, the filtration membrane is porous, thin, and negatively charged. Ooh, I'll address the negatively charged in a little bit, okay? That's going to be very, very helpful. But porous makes sense, right? What use is a filter if it's not porous, if things can't get through it? Thin, right, makes it nice too because it can move through with relative ease. So when we're talking about the filtration membrane, you will see we have a couple different layers there. There's 3 parts to it, right? I'll go through the parts real quick. The endothelium of the glomerulus, which are the capillaries, that's the simple squamous epithelium, right? You know what endothelium is. Then we have the basement membrane, because remember, epithelium sits on a basement membrane. And then the third is going to be the visceral layer of the glomerular capsule. So let me show you real quick. That's the view I want. My drawings are horrible. Real fast. So the visceral layer, right, of the glomerular capsule, you see how it's 2 layered here? It's this inner layer right here. That's the visceral layer. It sits right on top. All right, I'll show you a better picture here. Okay. So we're going to move filtered materials from your bloodstream into that space there. So remember in lab on Tuesday, I told you that there's 3 types of capillaries, okay? And it goes from hardly leaky to very leaky, okay? So this is the middle one. This is the fenestrated capillaries, not the sinusoidal, because those have these big gouges in it and stuff can leave the capillaries much easier. The fenestrated look like this. See those holes there? Those are the fenestrations. So it allows things to pour out through that area there. Right? Certain things can diffuse right through the endothelium, right? Like respiratory gases, right? Anything that's small, nonpolar, lipid soluble can just filter right through or diffuse right through the endothelium there, right? But in this case, the fenestrations are going to allow, right, certain dissolved substances to pass there. Note this. Large structures are restricted. Any of the blood-formed elements, things that you shouldn't see in your pee, okay? White blood cells, platelets, red blood cells, any of those big things, all right, should not be, okay, in your pee. All right. The basement membrane. Do you all remember what glycoproteins are and proteoglycans are? They're large molecules, all right? They're large molecules, but here's the important thing. They are large, negatively charged molecules. Proteins are usually going to have a negative charge. Do y'all remember, right, back in chapter 12, when you were learning about the neuron and you learned about the membrane potential, how it was positive outside and negative inside? Do y'all remember what gave the inside of the cell its negative charge? 2 things, primarily. Well, it's proteins and phosphate molecules. That's what made the inside of your cells negative. So proteins have a negative charge affiliated with them. right? So these are proteins, okay? So they're negatively charged, and that brings us up to this part, a negatively charged structure, because if something is negatively charged and it's going to come in contact with these negatively charged molecules, what's going to happen? They're going to repel from each other. Yep, remember? Opposites attract, but likes repel. So that's going to help us in certain situations. So that's going to help us because, all right, with the basement membrane, where am I going with this? Those plasma proteins, remember what I said? Proteins are negatively charged. So the negative charge of the basement membrane is going to help repel and push away, right, negatively charged circulating plasma proteins, which is nice. We can keep that stuff in the blood. We don't want to pee it out. It's very, very beneficial that way. All right. And then finally, our visceral layer of the glomerular capsule. This is the outermost, and you're going to see these cool cells here called podocytes. And so they have these processes called the pedoceles, and they're going to essentially create these slits on top of the capillary there. And the nice thing is, all right, these cells can contract or relax. And so they can manipulate the size of those slits, those thin spaces in between, all right, those pedoceles, and that can also affect what can pass through the filtration membrane. Right? So it's going to obviously keep a lot of the big molecules out. We might get some of the smaller proteins coming out. But for the most part, intermediate size, like the medium sized ones or the large proteins, those we do not want coming out. We do not. And then there's another cell here, which we'll get into later on when we talk about blood pressure, the mesangial cells. Right? The mesangial cells kind of just hang out. They're phagocytic cells, which means they just eat up the garbage that's lying around. But here's the thing, two things. Well, three things actually. They're phagocytic. So if something makes it through the filtration membrane, or if it partially makes it through the filtration membrane and it gets stuck, no problem. Mesangial cells come along, they chew it up, and they clear the debris from our filter. Much like if you're going to your pool, all right, and you're emptying the filter, you yank out the leaves in the water filter. That's what the mesangial cells are going to do. They're contractile, which is going to be very helpful because they're going to, we'll see here shortly, they're going to manipulate, all right, the size of these capillary slits and determine how much or how little material fluid can be filtered out. And then finally, we're going to be talking about these signaling properties here, all right, just briefly, not so much, but the contractile property I think is much more important here, right, when we get into the blood pressure. So just more on the mesangial cells later on, just kind of keep that in the back of your head there. So here you can see, all right, here's our blood capillaries, right, with the basement membrane. We'll zoom in here so you can see it a little bit better. But you can see on the outside, I just wanted to show you, right, there is the podocyte. It's got these extensions that come off, right? And then you have off of those extensions, these little pedoceles here. And so in between the pedoceles are those filtration slits. And so the podocyte can manipulate, right, how much space is in between those slits, which will then help to determine or affect what's getting filtered or what's not getting filtered here. So what we're going to do is we're going to zoom in now and take a closer look here at the filtration membrane. So you can see here are the three layers, right? There's the endothelium, there's the basement membrane, and then here are the pedoceles from the podocyte on the visceral layer. All right, so here's the thing. This is what you will see, all right, that's pretty much going to be able to move, right, through our filtration membrane. And if you look, they're small: water, glucose, amino acids. Those things are tiny. Various types of ions. Urea is a fluid there. All right, so the list goes on and on. All right, so these things can make it through. But if you look on the left side of our screen here, you think that leukocyte's going to make it out? I mean, even if it tries to go into diapedesis, it's still not going to be able to squeeze out through that. All right, so our red blood cells, our white blood cells, okay, and our large proteins, they should not be able to leave the bloodstream there by design. In a healthy individual, these things, yes. These things, no. You kind of see what I'm going with there? All right, so keep in mind, the endothelium, it's fenestrated. So it's going to restrict large molecules from making it through. Then we have our basement membrane, right? That's going to block a lot of the large proteins because remember, right, those glycoproteins and those proteoglycans, those are negatively charged molecules that sit right there in that basement membrane. So their negative charge are going to repel anything that's negatively charged here, hopefully. Right? And then we've got our podocytes here with the pedoceles, which they can then manipulate the distance and spacing in between, and they can let certain things in and out. Okay, so look at that stat. Your kidneys are producing 180 liters a day. Okay, now you're not peeing that out, okay, because a lot of that gets resorbed back into your bloodstream, right? But that's a lot, okay? That's a lot. So that's what's getting filtered out. So, all right, so it's primarily going to be, all right, solutes. A lot of those solutes will be traveling out through the blood plasma that gets filtered out. Okay. So whatever that does not get filtered out of our glomerulus just stays in the bloodstream. And maybe it'll get filtered out in another nephron, all right? Or it might get filtered out in its next pass, right? So whatever doesn't get filtered out stays in the efferent arteriole and circulates through, all right? And we're going to talk about in lab today, all right, the blood flow through our kidneys today. And again, if anything gets trapped, okay, we got our mesangial cells to clean it up. They're going to clean out our filter like the pool guy. Cool. I know it's not the most exciting stuff. Wait till we get to the blood pressure stuff. I think you might like that. So you should pay really close attention to this slide. Wink, 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 wink. All right. All right. So there are 3 categories of substances that are found in your blood. Okay. You got freely filtered, not filtered, and then limited filtration. So freely filtered, and this makes sense, okay? Really small substances. So all of those things right there should make sense to you. Water, glucose, amino acid, ions. Right? They are very small, so they'll pass right through the filtration membrane. Things that cannot make it out. Large blood-formed elements, all right? White blood cells, red blood cells, platelets, nada, and our large proteins. They won't make it through. Now, limited filtration, here's the one that, kind of varies here. Right? So proteins that are of medium size or what we call intermediate sized, right? Normally, they should have a harder time. And so depending on what's going on, you might get some filtered out. Like if you have high protein levels going on, if you're on a high protein diet, you're going to see, because it's happened to me. I was a guinea pig in a past life. No, I was a guinea pig for asthma research studies. That's the nice thing about being a poor college or grad school student. You found ways to make money. I sold plasma. I did it all. So I used to do these research studies for an asthma group, and I would take these drugs. Good news is all these drugs are on the market now. So, all right. And I think that I am not affected, all right, too much. Okay. But what we used to do is at the time, I used to be very, what's the word I want to use? Well, active. And so I was on a high protein diet. I was drinking like 4 protein shakes a day. I recommend that you do not do that, all right? Because my blood pressure was increasing, all right? Because if you increase protein, you're increasing your oncotic pressure. And what does protein do? It draws things into your bloodstream, okay? So it increases your blood volume. But point being is, when I used to do my urinalysis, they're like, you got proteins in your urine and you got a lot of them in there. So they were concerned about my kidneys for the medication. And I said, well, I'm taking a lot of protein. They said, okay, cut it out for 2 weeks and let's just see if that's still happening. I stopped it for 2 weeks, no problems after that. All right. But point being is, that's what will happen if you're on a high protein diet or some other pathological things that could occur, like rhabdomyolysis, your muscles breaking down, right? But usually the intermediate or medium-sized proteins are going to be blocked for 2 reasons. 1, because of their size, and 2, like I said, that negative charge that comes from the basement membrane. Right, so you really shouldn't see it there, but again, you can see, all right, intermediate-sized proteins depending on the situation there. Here we go. We're going to move on into blood pressure now, y'all. Okay. So first of all, we're going to talk about, there's a couple different pressures that are associated, right, with what we call glomerular filtration. Glomerular filtration, you tell me what that is. Well, what's the glomerulus? Capillary, the loop of capillaries. And so what's the filtration process? What are we talking about? It's going into the capsular space. Yeah, that's what I'm assuming that you meant, right? So that's when we talk about glomerular filtration, we're talking about the stuff leaving your capillaries and going into the capsular space. What we just talked about, that filtering process there, right? So that's what we're going to talk about here. So there's a pressure associated with that, right? And so you heard me say it, right? Things didn't change for the circulatory system. This is that hydrostatic pressure, right? What pushes, that push pressure on, right, the walls of the vessels there of the glomerulus. They're just capillaries, right? So that hydrostatic pressure pushes water and any of those solids that are in your blood plasma out of the glomerulus and into the glomerular space, into Bowman's space there, right? And then it gets, all right, captured and put into the proximal convoluted tubule. So if you look here, all right, this is an important thing. This pressure is going to be higher than your systemic blood pressure in your other capillaries. Because remember, the blood pressure in your capillaries would go from 40 millimeters of mercury from the arterial end of the capillary bed to 20 millimeters of mercury at the venule end of the capillary bed. That should kind of sound familiar to some of you, okay? So what we're saying is it's a little bit higher than that. So we're ensuring that the filtering process is occurring. That's our goal. We want to get stuff filtered through, right? So we have the filtration process to occur. So we're just going to increase the pressure there. So that's going to happen. And this is done by, and I mentioned this in lab, remember I said that the afferent arteriole has a larger diameter than the efferent arteriole. So if you have a larger diameter, you got a larger lumen, you have less resistance, right? Increased flow. Smaller diameter, smaller lumen, increased resistance, decreased flow. Okay, that setup there is going to help contribute to that increase in filtration pressure there. Almost kind of backs up the blood. Now, I don't want you to think it becomes stagnant, all right? But it's going to keep it, all right, where it's going to be able to increase the pressure so we can push it out of the glomerulus and into the capsular space. All right, so here we go. So that first pressure that I didn't mention here, I should have just pointed that out to you. That hydrostatic pressure, HPG, that's what that, you all remember those letterings there, okay? HP, hydrostatic pressure, and the small g, the subscript there, is for the glomerulus, okay? So this pressure should be the highest. Now we're going to talk about these two pressures that oppose the hydrostatic pressure in the glomerulus. So we can't get away from the colloid oncotic or colloid osmotic pressure. Because why? Yeah, proteins. Okay, we're going to have proteins in our blood, right? So we can't get away from that. So y'all recall, right, your blood, because of the proteins, the plasma proteins, are going to have that pulling, all right, they're going to try to pull things into the bloodstream, right? And so here we're going to have, right, our colloid osmotic pressure in the glomerulus, notated as OP subscript G, G for the glomerulus there, right? And so again, this is going to oppose filtration. And then the other one here is the capsular hydrostatic pressure, notated as HP subscript C. And so as fluid moves into the glomerular space there, okay, that fluid there is going to exert a pressure on the outside of the capillaries. And so because of that, that's also going to oppose filtration, right? But it also impedes movement. Law of physics. Cannot have two things occupying the same space. If there's fluid out there in the glomerular space, it's hard to move more fluid into that space because there's already a physical presence of some matter there, okay? All right. So there's an equation for this, of course, which you all need to know. And there it is. This equation, commit this to memory. We're going to talk about the net filtration pressure. Right? So the next concept is going to be net filtration pressure. And we've discussed net filtration pressure in the circulatory system. Right? When things were moving out of our capillaries into our tissues, and when things were moving from our tissues into the capillaries. And so that equation there, net filtration pressure, is equal to the hydrostatic pressure of the glomerulus minus, all right, the two opposing pressures, the sum of the two opposing pressures, which is the oncotic or osmotic pressure in the glomerulus and the hydrostatic pressure outside of the glomerulus in the capsular space there. So you can see the example here. If we measure the hydrostatic pressure inside of the glomerulus, it's 60 millimeters of mercury in this example. And then we measure the osmotic pressure inside of the glomerulus. It happens to be 32 millimeters of mercury. And then we measure the hydrostatic pressure in the capsular space there as 18 millimeters of mercury. We sum those up, we add those together. That's 50. And then we just take the difference, right, from the hydrostatic pressure of the glomerulus and then the opposing pressures. That gives us a positive value. So if we have 10 millimeters of mercury, that favors filtration, just like we saw before, right, in our circulatory system. So it's like it says here, if the pressures promoting filtration are greater than the opposing pressures there. Okay, that's what we're looking for. The only way for filtration to occur is if our the hydrostatic pressure is higher than the sum of the opposing pressures. Exactly, which we really shouldn't see, but there'd be a problem. We would be having it would be not good. Not good. Because a lot of times too, if that occurs, you can get a buildup of metabolic wastes in the bloodstream. If you can't get rid of it, then you're going to have a whole host of issues going on. Okay. You get end organ damage, tissues can eventually you get toxicity levels, and then a whole host of things happens. Coffee. Okay, so that's what this picture is showing us. Okay, you can see the hydrostatic pressure inside the glomerulus pushing out, and then our two opposing pressures, the osmotic colloid pressure, which is going to be that pulling pressure, right? Because the plasma proteins are going to be trying to suck fluid back in. And then we have our hydrostatic pressure that's exerted on the outside of the glomerulus because of the fluid that's in the capsular space here. So these two are the opposing pressures. We take the sum of those two and then take it, all right, the difference between those two values, which will give us hopefully a positive value that favors filtration. And again, you can see here, I'm going to point random things out to you. Look at the lumen here, the diameter, right, of the afferent arteriole, and then look at the lumen size of the efferent arteriole. Bigger, smaller, more blood flow. That's going to help contribute to the hydrostatic pressure. Because we're going to talk about now how the size of this thing will affect some of these values here. All right. So now we're going to be talking about variables influenced by the net filtration pressure. So there's another pressure there we're going to talk about called glomerular filtration rate. So again, glomerular filtration rates, right? Anytime you see rate, like heart rate, we're involving time, okay? So glomerular filtration rate means we're going to involve two things, volume and time, right? So the definition is the rate at which the volume of filtrate is formed. And of course, we're going to always use our favorite 1 minute as our unit of time here. So in the period of a minute, we're going to see how much filtrate is formed, right, from the glomerulus there. So some things here that we're going to notice when we have an increased net filtration pressure. So we increase our net filtration pressure, which is what? This right here. This whole story. Right. If we increase our net filtration pressure, we're going to see an increase in the glomerular filtration rate. Makes more sense, right? We have more blood coming into the area. We're going to be filtering out more. Right? We're also going to see, right? Because an increased net filtration pressure, we're going to see more solutes in water that are going to be in the tubular fluid because we're moving more of that fluid out into our proximal convoluted tubule. And also, we're going to increase urine production. So the first three things we're going to see an increase. But what we will also see is a decrease in filtrate reabsorption. So less stuff is leaving the tubular system and going back into the bloodstream. Hence, that also increases our urine volume. And we're going to go through some things here, but for right now, just kind of commit some of that to memory there. Right, so look at that top statement there. GFR is tightly regulated. Okay, so no matter what's going on in your body, your blood pressure could be fluctuating to 180 over 120, or it could be swinging down to, well, I won't go that low. It could swing down to 70 over 50. But your kidneys are still going to try to maintain the glomerular filtration rate at a specific level, right? At a specific level. And so what we're going to see, right, because certain conditions can be occurring in your body. So we might have to alter, right, urine production, for example. Right? So if you're dehydrated, do you think you're going to be peeing a lot? No. But if you're really hydrated, like you just drank 2 gallons of water, you're going to be peeing a lot, right? So that's what we're going to do here. We're going to, depending on what type of physiological conditions are going on, right, is going to determine your urine production here. And so here, when we're talking about the glomerular filtration rate, there's a couple ways that we're going to influence that or affect it or change it. Right? The first thing, and I mentioned this before, is that we can make the diameter of the afferent arterial bigger or smaller. Will affect the flow of blood into the glomerulus there. Another way to affect, right, our GFR, how much fluid is being pushed out into the capsular space, is we'll mess around with the filtration membrane. We'll make it easy for stuff to get filtered through, or we'll make it hard for stuff to get filtered through. So there's two ways to do this. We have intrinsic controls, which means the kidney is going to do it all by itself. And then we have extrinsic controls, things that are going to be outside of the kidney. Endocrine system. We're going to get some hormones in on this thing. Okay? Remember our friend aldosterone? And remember our buddy ADH or antidiuretic hormone? That's going to be some of our extrinsic controls there, which we're going to talk about. Okay, we learned about what they did in the endocrine system, that they just make the kidney do something, increase urine, decrease urine. Now we're going to see how it specifically is able to do that. So let's start off with the intrinsic controls here, right? So for the next few minutes, as I talk about this, I want you to keep in the back of your mind that the kidney is always going to want to maintain, right, your glomerular filtration rate at a certain level. Okay? It wants to keep it, best way I can say it is constant. Okay? Constant. So we're going to see how the kidney is going to do this, regardless of what's going outside the kidney. So you can change your systemic arterial pressure or blood pressure, all right? But the kidney is going to try to maintain its glomerular filtration rate, right? And we're going to talk about what happens, right? Like, for example, if your systemic blood pressure goes up, how the kidney is going to make sure, all right, that it's going to still produce the same amount of filtrate, regardless of what's going on outside, all right? We're going to talk about that. All right, so we have two mechanisms. Myogenic, that should look really familiar to you. Okay, that means there's some muscle involvement going on. And then we're going to talk about our tubular glomerular feedback mechanism, which is, we briefly talked about the juxtaglomerular apparatus there. We labeled it in lab, but now you get to see it in action and what's going to happen there. So let's start off here with the myogenic response. If it's going to be muscles, there's only going to be one type of muscle that we're going to really be dealing with here, and that's going to be smooth muscle. And so we know where our smooth muscles like to hang out, in blood vessels. So what blood vessel? Specifically, the afferent arterial. So that's what the myogenic response is going to be. We're going to either see your smooth muscles contract or they're going to relax depending on the amount of stretch that occurs, whether it's a decrease in blood flow or an increase in blood flow. So let's start off with our first example. Your blood pressure starts to go down. Okay, we have a decreased blood pressure, and we know this because we talked about this mechanism when we talked about the circulatory system and blood pressure. So when you have a decrease in blood pressure, what's that going to do to the stretch on the vessel walls? It says it up there. It becomes less, right? The baroreceptors are going to have less of a stretch there. So what happens then if there's less of a stretch because your blood pressure is decreasing? Remember, we want to maintain a constant glomerular filtration rate. We want to still be able to produce 180 liters, right, of filtrate a day. So that means what we need to do is we got to get more blood to flow through there. So the smooth muscle cells will relax. That causes the lumen of the afferent arterial to increase through vasodilation. That allows more blood to enter into the glomerulus there. And then we'll see we maintain that glomerular filtration rate. Because if you have a decreased blood pressure, you're going to see a decrease in blood flow. So now let's talk about the other. Come on, there we go. What happens? We have an increase in blood pressure. Really salty meal or something. Okay, so your blood pressure increases. That means you're going to see more of a stretch on the blood vessel walls. So they're going to respond to that by causing vasoconstriction. That vasoconstriction from the smooth muscles is going to then decrease the flow of blood into the glomerulus, right? Because there was a sudden increase. And so, again, the final outcome is to always keep things constant. So essentially what you're seeing is, for example, with both of these situations is you'll see an initial, like for this, let me go back. For this one, you'll see an initial decrease in blood flow because the blood pressure went down. So in order to increase that blood flow, what do? We decrease the resistance. How do we decrease the resistance? We increase the size of the lumen. All right, so blood can start flowing again. Okay, it's like pinching off the hose, right? You bend the hose, your water flow decreases, right? But then when you take the bend away, it starts flowing again. And so that's what we want to see here. Remember, our end outcome is always to keep the glomerular filtration rate normal because your kidneys are shooting for 180 liters of filtrate formed a day. That's their goal. That's what they're trying to do. Same thing here. So when you get an increase in blood pressure, you'll get a quick surge, right? An increase in the flow of blood. So we want to tamp that surge down so we cause vasoconstriction. And so that decreases the blood flow, all right, into the glomerulus or gets it back down to its normal level there. So we can always keep this constant. So it's a compensatory mechanism is what we're really seeing. We're trying to compensate. So this picture here, right, figure 2413, right here, this is what we're seeing here. Decrease in systemic blood pressure. So initially, blood flow decreases for a little bit. Well, we got to fix that. We can't have less blood coming in. So we just vasodilate this blood vessel that allows more blood to come in. And then over here, we have an increase in blood pressure. So you get a quick surge here of increased blood flow, right? So then we're going to cause vasoconstriction to decrease that back to its normal amount so we can then get more or maintain that glomerular filtration rate. 180 liters. All right, so that's the myogenic response. So our backup system is our tubular glomerular feedback mechanism here. right? Now, this is going to be for increased blood pressure here, right? So what we're going to see is, right, for the blood pressure, when it increases here, we're going to see more salt. So you have, right, the cells there, remember the macula densa cells that were located in the distal convoluted tubular, all right? So these cells are going to monitor, right, the content of sodium chloride, salts. And so what we're going to see, if there's an increase in salt, what does that mean? We have an increase in what? What's usually associated with salt? Yeah, water. Remember, water likes to follow solutes. It's the follower. So if you have an increase in the amount of salt in the tubular system, you're going to have an increase in the amount of water, which means you're going to have an increase in blood volume, which means you're going to have an increase in blood pressure. All right, so in this case, what we're going to do is we're going to create vasoconstriction. So we're going to decrease the lumen of the afferent arteriole even more, increasing the resistance, decreasing the flow. All right. So I want to say, I do have a picture here. All right. I want to show you this picture first. This is kind of showing you, I remember our 180 liters a day. So this is pretty much what the intrinsic mechanisms or factors are going to be able to handle. But eventually, you're going to get to where they're not going to be able to handle, all right, maintaining this value here. You can only vasoconstrict or vasodilate the afferent arteriole so much. Right? That's where we're going to bring in some other thing, the extrinsic factors here, all right? But that's what I wanted to show you. So this is what this slide is talking about here, the limitations that we're going to be dealing with here. We're going to be, this range here to maintain that normal pressure is going to fall between 80 and 180 millimeters of mercury. So when we fall below that value, like I said, we can only vasodilate so much. Then what we're going to do is we're going to then change, right, our fluid volume. So if our blood pressure falls so low, okay, one of the factors that influences our blood pressure is blood volume. We know heart rates and all that, and we know the total peripheral resistance plays a role, but we're talking about the kidneys here, okay? So our kidneys can affect blood volume. So we're going to talk about only what they can control. So when your blood pressure goes below 80 millimeters of mercury there, you're at max dilation for the afferent arterioles, okay? And our blood pressure is still going down. Then what we're going to do is, because remember, we want to keep this constant, but now this starts to fall. The glomerular filtration rate starts to fall, and our glomerular blood pressure has fallen. So here's what we're going to do. We're going to try to maintain our blood volume as best as we can. So we're going to just pretty much decrease significantly urine formation or stop it altogether, so we can retain that water, so we can maintain or contribute to the blood volume there. Now, if our blood pressure gets way high, above 180 millimeters here, same thing. We've reached maximal vasoconstriction in the arterioles there. They can only constrict so much, all right? But yet, we're still going up in the glomerular filtration rate because there's just too much blood coming in, right? And the pressure has increased. Well, guess what? We've got to do, the only thing the kidneys can do is affect that blood volume. So we're just going to start making a lot of urine. We're just going to start drawing fluid out of our blood, namely water, because that's 92% of the blood plasma. So we'll just start pulling water out of your blood and just putting it into the tubular system and making urine there. And hopefully that will help, all right, to handle this situation of the increased blood pressure there. So like I said, there's limitations to what we're dealing with here. Kind of sucks, but that's just how it is. All right, those are the intrinsic methods there. I want to get into the other ones, the extrinsic, because I think you folks will kind of enjoy that. All right, the extrinsic factors or controls. right? Hormones and our nervous system are two control centers here. So now, okay, remember the intrinsic is there to maintain the glomerular filtration rate at 180 liters of filtrate made a day, okay? But now those values have changed. We're running into this situation. We're running here where we can't maintain this value. It's either starts to drop or it starts to increase. So we're going to call in the big guns. We're going to call in our extrinsic factors here, right? Because we need to change our glomerular filtration rate because the intrinsic couldn't do it. All right, so let's talk about the neuron, the nervous system division that can help out. Sympathetic, okay, fight or flight, right? So fight or flight, remember, sympathetic nervous system is the only division that can affect blood vessel diameter, right? Parasympathetic doesn't do jack with that, right? So that means we're going to cause vasoconstriction, right? So here we have a situation for a decrease in glomerular filtration rate, right? We need to decrease the glomerular filtration rate. So how do we do that? Vasoconstriction. In both afferent and also the efferent here. Now, our hormonal system is going to kick in here too, right? Because our blood pressure is increased, we want to decrease it. So we've got renin here that is going to help us out with that situation. So y'all remember what renin does? Do y'all remember what renin does? Well, the answer's up there. All right. Renin helps with the formation of angiotensin 2. Okay, so remember, your liver is always making angiotensinogen inactive all the time. So you're sitting here, you're making it, it's dumping into your blood. Renin converts angiotensinogen into angiotensin 1. Then it goes to the lungs. With the lung ACE, the angiotensin converting enzyme converts it into angiotensin 2. All right, so all that happens. So what does that do? Remember I told you we're going to revisit these cells? The mesangial cells come into play, and they are going to contract, and they're going to decrease, right, the ability of the filtration membrane to filter stuff out. And by doing that, less stuff is leaving your blood and staying in the blood. You're going to increase your blood volume, therefore increasing your blood pressure, which is then going to help increase, all right, your overall blood pressure there. All right, and if you think about it too, the sympathetic nervous system here is also going to help us with water conservation to maintain, all right, fluid electrolyte levels, things of that nature. All right, now how do we increase the glomerular filtration rate when we're dealing? Remember this guy, AMP, atrial natriuretic peptide? That's that hormone that's released, right, in the atria when you have an increase in blood volume. More fluid is coming back to the atria, puts a greater stretch on the atrial walls. AMP gets released, and now we're going to talk about how AMP is going to then, well, the overall effect of AMP is to do what? Is it up there? No, it's not. What does AMP do? It's going to decrease the blood pressure because, well, it's an increase in blood volume. Increase in blood pressure causes its release. It's all right. It's all right. You're learning. Right? So here's what it's going to do, all right, for our GFR. Okay. It's going to increase the GFR because remember, if we increase GFR, we're going to increase our urine production. If we increase urine production, we're going to decrease blood volume. Right? If we decrease blood volume, we decrease blood pressure. So how does this work? Okay, so AMP is going to affect the afferent arteriole, and so it causes vasodilation of the afferent arteriole, and so that allows more blood to flow into the glomerulus so we can filter more filtrate out of your blood, increasing urine production. But it's also going to take renin off the board. Because if we can't produce renin, we cannot produce angiotensin 2. And so it inhibits the production of renin, And it also causes the mesangial cells to relax, loosening up its grip on the filtration membrane. If the mesangial cells are relaxing, then we can filter more stuff out. And so that allows us to increase our glomerular filtration rate, which allows us to increase the amount of urine production, decreasing fluid volume or blood volume, therefore decreasing blood pressure. Remember, the kidneys can only really contribute to the regulation, right, for blood pressure dependent on fluid, right? How much fluid you're retaining or how much fluid you're getting rid of. That's its contribution there. Okay, so net increase in glomerular filtration rate is going to cause more urine being produced, which means you're going to pee that fluid out that used to be in your blood, and that's going to decrease the blood volume, which causes a decrease in blood pressure. I know it's a lot to throw at you. I don't expect you all to get this right away, but you go over it a couple times, hopefully you'll understand. And I'll even throw a picture in here for you. Maybe that'll help. All right, so that's what this is showing you. Decreasing glomerular filtration rate through the sympathetic nervous system here. All right, so here you're going to see there's two ways to do it, okay? So the sympathetic nervous system is going to cause vasoconstriction here of the afferent arteriole, which means less blood. Okay, we cause vasoconstriction, we decrease blood flow, right? Less blood is going into our actual glomerulus. And then also here, whoops, we're going to see, all right, the granular cells there that are going to produce renin, right? Renin causes the production of angiotensin 2. And then that angiotensin 2 is going to cause the mesangial cells to contract. And so they're going to decrease the surface area here of our filtration membrane. Everything kind of just clamps down. So less stuff can leave the glomerulus. If less stuff is leaving the glomerulus, where is it staying? If it can't leave the blood, where is it staying? In the blood. Okay, so less fluid leaving the blood, we're increasing our blood volume, increasing our blood pressure. But do y'all remember what angiotensin 2 also does? It also affects the production of aldosterone and ADH, which we'll talk about, well, not today. We don't have time. Okay, so again, look at this picture here. This is figure 2414A. And all right, it'll kind of look through it, and at the bottom here, it kind of walks you through, all right, the results and the net effect there. It's a lot. It's almost like a story. You kind of got to go through it a couple times. It's not easy. No one said it was going to be easy. Mama said there'd be days like this, right? So here, now we're going to see an increase in the glomerular filtration rate with our other hormone called AMP, Atrial Natriuretic Peptide. Okay, so here's what sets up the release for atrial natriuretic peptide, an increase in blood volume, which is meaning you have an increase in blood pressure, right? So your blood pressure is increased because you have a larger blood volume. And so AMP is going to cause vasodilation here of the afferent arteriole. Look at that thing. It's huge. So more blood can flow into the glomerulus there. So that's going to promote filtration. So more fluid is going to come out. That's one way to decrease our blood volume. The other way is going to be, right, we're going to inhibit the release of renin. If we're not releasing renin, we're not making angiotensin 2. Angiotensin 2 causes the mesangial cells to contract and decrease the surface area here of our glomerulus. But since we're not making that, the mesangial cells have to relax. Look at the size of this thing compared to the other one. This one's all small. This one's nice and big. So our surface area is grown. So more things can leave our glomerulus, more fluid leaves. So it means we're going to be forming more urine, which means we're going to be decreasing our blood volume, which means we're going to be decreasing what? Blood pressure. Good. I know it's not easy. All right, this actually is a good place to stop. I don't want to, but this is a good place to stop. Okay. Let's stop here. It's a lot. I know it's a lot. Good thing that you have the weekend to learn it and go over it. But I promise you,